This presentation will explore how to effectively and efficiently protect biodiversity hotspots of vulnerable and endemic species. The issue is how to conserve biodiversity in the U.S. To start, there is a mismatch between lands that are protected and areas of vulnerable hotspots which do not overlap. The most vulnerable species are endemics, or those with a small range size, and these are species that will benefit the most from protection. So how well do we currently protect endemic species? Only 7.8% of the lower 48 is within a protected area. As shown in Figure 3b, most protected areas are in the west, where many of the areas are federally owned, while endemic richness is centered in the southeast U.S., shown in Figure 1, where much of the land is private or not protected. The authors say that this is where the most protection needs to be. The authors, Jenkins et al., weighed the species by the percent of protected range size to determine which areas to conserve, and Brown et al. Replied to declare that this is not the most effective or efficient way to conserve biodiversity. They defend this claim with 4 lessons in conservation planning. First, setting priority areas involves more than determining how many vulnerable species live in the areas that we are able to protect. Next, that conservation planning involves actions, not identifying areas able to be protected. Third, that conservation plans need to take into account the economical, political, and social inhibitions on the planned actions. And finally, conservation plans involve conserving all species, not only vulnerable ones. The authors suggest that Jenkins et al. Consider these criteria. This is something to keep in mind as we continue on to our research question. What areas should be protected in order to efficiently but effectively conserve biodiversity? We use GIS models to identify the concentration of protected areas and the density of endemic species. This map shows the distance of any given protected area to the nearest protected area. It shows that there's a sparse distribution of protected areas in Texas, as well as in the Southeast and Midwest. We compare this to the next map, and see that this map shows a high density of endemic species in the southeast. Since there is a mismatch between the two, it's very important to protect biodiversity in areas where endemic species and protected lands overlap. Even if the proper measures are taken to conserve these protected areas, factors like urbanization can influence the execution of a conservation plan. This group was also interested in using the overlapping range of a vulnerable species in protected areas. They used survey data of a butterfly that is endemic to the Rio de Janeiro state of Brazil to create a predicted distribution map, which overlapped with 19 protected areas. However, solely because of urban areas, the effective distribution was reduced to 8.7% of the predicted model. Only 2 of the observed occurrences that overlapped with protected areas were predicted. This group determined that the most effective and efficient way to conserve this species is to breed it in captivity. However, species don't occur at all locations within their range, and richness patterns may differ between range maps and survey data. Figure 1 shows the range map of a southern Africa bird in gray, the observed in red, and not observed in blue at a resolution of 0.25, 0.5, 1, and 2 degrees for A, B, C, and D. The ratio shows the surveyed occurrences per total number of well-surveyed cells falling within the species' geographic range. Applying this example to protecting biodiversity, using a zoomed-in range map with a coarse resolution to make conservation decisions will lead to a scale mismatch. The authors determined that conclusions of species richness and other patterns derived from range maps of less than 2 degrees resolution may be misleading. This is something to keep in mind while researching biodiversity conservation plans. Thank you.